Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied about classification of amino acids, and now we are going to study the same further. Classification based on nutritional requirements. Essential or indispensable amino acids. The amino acids may further be classified according to their essentiality for growth. Thus, isoleucine, leucine, threonine, lysine, methionine, phenylalanine, tryptophan, and valine are essential amino acids. Their carbon skeleton cannot be synthesized in human beings, and so preformed amino acids are to be taken in food for normal growth. In this diagram, you can see different amino acids and their sources in the food like legumes, spinach, oats, pumpkins, avocado, cauliflower seeds, etc. Histidine and arginine are the semi-indispensable amino acids. Growing children require them in food, but they are not essential for the adult individual. In this diagram, we can see the food that contains histidine. They are fish, milk products, meat, dry fruits, and pulses. Here in this diagram, we can see that the food contains arginine. These sesame seeds, soya proteins, crab, shrimp, lobster, turkey, chicken, pork, beef, salmon, pumpkin seeds, and spirulina. Non-essential or dispensable amino acids. The remaining 10 amino acids are non-essential because their carbon skeleton cannot be synthesized in the body. However, they are also required for normal protein synthesis. All body proteins do contain all the non-essential amino acids. Naming or numbering of carbon atoms. Carbon atoms are in amino acids in sequence are named after letters of Greek alphabets, starting from the carbon atom to which carboxyl group is attached. In this diagram, we can see that C1 is a carbon atom where carboxyl and amino groups are attached. In this graph, we can see that a list of amino acids is given along with the three abbreviation, one abbreviation, letter abbreviation, and molecular weight. Properties of amino acids. Glycine, alanine, valine, serine, tryptophan, histidine, and proline are sweet in taste. Leucine is tasteless, while isoleucine and arginine are bitter. Sodium glutamate is a flavoring agent. Aspartame, an artificial sweetener, contains aspartic acid and phenylalanine. All amino acids have high melting points, that is more than 200 degrees centigrade. All amino acids are soluble in water and alcohol, that is polar solvent, but they are insoluble in non-polar solvents. Example, benzene. Ampholyte and isoelectric point. Amino acids can exist as ampholyte or Zwitter ions. A German word Zwitter means hybrid in solution depending on the pH of the medium. The pH at which the molecule carries no net charge is known as isoelectric point. or isoelectric pH or PI. In this diagram, we can see non-ionized form of amino acids above, whereas sweeter ion form is shown below. In such acidic solution, they are cationic in form, and in alkaline solution, they behave as anions. At isoelectric point, the amino acid will carry no net charge. All the groups are ionized, but the charges will cancel each other. Therefore, at isoelectric point, there is no mobility in an electric field. Solubility and buffering capacity will be maximum at isoelectric pH. To such a solution, if we add hydrochloric acid drop by drop at a particular pH, 50% of the molecules are in cation form and 50% in sweeter ion form. This pH is known as pK1 with regard to COOH. If more HCl is added, more molecules become cationic in nature and solubility increases. On the other hand, if we titrate the solution from isoelectric point with NaOH, molecules acquire the anionic form. When 50% of molecules are anions, that pH is called pK2, that is with respect to NH2. In the graph, we can see that glycine has pK1 value of 2.34 and pK2 value of 9.6. The isoelectric pH, that is PI, for monoamino monocarboxylic amino acids has been calculated. PI is equal to pK1 plus pK2 divided by 2. For example, PI of glycine is equal to 2.4 plus 9.8 divided by 2, which gives 6.1. In this table, we can see that a list of amino acids associated with their pK1, pK2, pK3 and PI is given. It is evident that the buffering action is maximum in and around pK1 or at pK2 and minimum at PI. In the case of amino acids having more than 2 ionizable groups, correspondingly there will be more pK values. For example, aspartic acid. From these values, it can be seen that at physiological pH of 7.4, both carboxyl and amino groups of amino acids are completely ionized. Thus, to be very correct, the ionized forms are to be shown as structures of amino acids. The pK value of imidazolium group is 6 of histidine is 6.1 and therefore as a buffer at the physiological pH of 7.4. The buffering capacity of plasma proteins and hemoglobin is mainly due to the histidine residues. Optical activity. Amino acids having asymmetric carbon atoms exhibit optical activity. Asymmetry arises where 4 different groups are attached to the same carbon atom. In this diagram, we can see that D-alanine and L-alanine are optically active at C1. Glycine is the simplest amino acid and has no asymmetric carbon atom and therefore shows no optical activity. All other are optically active. The mirror images form forms produced with reference to the alpha carbon atom are called D and L amino isomers. In this diagram, we can see that glycine has C1 cannot be asymmetric carbon since it has two hydrogen atoms attached. The L amino acids occur in nature and are therefore called natural amino acids. T amino acids are seen in small amounts in microorganisms and as constituent of certain antibiotics such as clamicin S, polymyxin, actinomycin D, and varenicomycin, and also bacterial cell wall peptidoglycans. Isoleucine and threonine have two optically active centers and are therefore each has 4 diastereomers. In this diagram, we can see that isoleucine has two optically active centers leading to 4 diastereomers. Sorensen's formal titration. Amino acids cannot be exactly titrated. It is if, for example, 1 ml of 1 normal solution of glycine is titrated against 1 normal sodium hydroxide, the alkali requirement will be less than 1 ml. This is because hydrogen ions released by ionization of carboxyl group are partly taken by the amino group. To circumvent this problem, excess formaldehyde is added to the solution, which converts amino group into neutral amino dimethyl derivative. Therefore, titration can be completed at the end point. In the above example, after addition of formaldehyde, exactly 1 ml of 1 normal sodium hydroxide is utilized in the titration. General reactions of amino acids. Decarboxylation. The amino groups will undergo alpha decarboxylation to form the corresponding amine. Thus, some important amines are produced. For example, histidine forms histamine plus carbon dioxide, tyrosine forms tyramine plus carbon dioxide, tryptophan forms tryptamine plus carbon dioxide, lysine forms cadaverine plus carbon dioxide, whereas glutamic acid forms GABA plus carbon dioxide. In the diagram, we can see that alanine is undergoing decarboxylation. Second is amide formation. The COOH group of dicarbolic carboxylic amino acids, that is other than the alpha carboxyl, can combine with ammonia to form the corresponding amide. For example, aspartic acid plus amine forms aspargine, or glutamic acid plus ammonia forms glutamine. These amides are also components of protein structure. The amide group of glutamine serves as the source of nitrogen for nucleic acid synthesis. In this diagram, we can see that a free amine undergoing amide formation. Reactions due to amino group. The transamination, the alpha amino group or of amino acid is transferred to alpha keto acid to form the correspondingly new amino acid and alpha keto acid. This is an important reaction in the body for the interconversion of amino acids and for synthesis of non-essential amino acids. In this diagram, we can see that two amino acids undergo going transamination to form two different amino acids. Oxidative deamination. The alpha amino group is removed from the amino acid to form the corresponding keto acid and ammonia in the body. Glutamic acid is the most common amino acid to undergo oxidative deamination. In the diagram, we can see that glutamate is with the help of enzyme glutamate dehydrogenase form alpha ketoglutarate. Formation of carbamino compounds. Carbon dioxide adds to the alpha amino group of amino acids to form carbamino compounds. The reaction occurs at alkaline pH and serves as a mechanism for the transport of carbon dioxide from tissues to the lungs by hemoglobin. Hemoglobin NH2 plus carbon dioxide gives carbamino hemoglobin. In this diagram, we can see that carbon dioxide with an amino acid forms carbamino compound. Transmethylation. The methyl group of methionine after activation may be transferred into an acceptor which becomes methylated. Methionine plus acceptor gives methylated acceptor plus homocysteine. In this diagram, we can see that methionine helps in transfer of sulfur. Here ends our session. Kindly like, share and subscribe the channel. Thank you.